Hello, I'm Bastien Chopard, and with my colleague Rémy Petkantchin and Simon de Meyer, I'd like to present some results we obtained about thrombolysis within the INSIST project. So INSIST stands for In Silico Trial for Ischemic Stroke, and it's a project funded by the European Union with several partners, university, research center, and company with different expertise. So what is the problem of ischemic stroke? It's illustrated here, where you have one of your brain vessel which is occluded by a clot, and as a result the blood is not coming to all of your brain, and it can result in very severe damages. It can go from health to mild to moderate disabilities. So fortunately there are some treatment for that. One is thrombectomy, which is a mechanical retrieval of the clot. It's a rather complicated procedure, and much less invasive is thrombolysis, where you inject a chemical, a molecule called TPA, that will dissolve the clot. So here you have an illustration of the brain vessel, and you have a clot that is occluding one part of the brain circulation, and as the TPA reaches that clot, there's a dissolution, and finally the circulation is restored and the patient is saved. So to study such a phenomena, we have to identify the main component, and one is about the flow, so blood flow that brings the TPA towards the clot, and this depends on the pressure drop across the clot and the permeability of the clot. Second is the lysis, success of the lysis, which is the ability of the TPA to actually dissolve the clot, and that depends on its composition. So unfortunately these parameters are not well known, and to study them we will separate them before we can put them together again to have a full picture of the process.
So once we have studied them and found a reasonable quantity for each one of them, we can then build a numerical model that would simulate the thrombolytic treatment in any given patient with any given physiological condition. So here in green you see the injection of TPA through the patient artery, which will then start to dissolve the clot, which is in red, and while doing so, flow will be restored in the patient vessel, and thus we hope that the patient can be saved through this treatment. So how do we build such a model? So you need to start with a set of basic experiments that are reproducible to observe the process that you want to study. You need to identify the essential features and the basic components that can reproduce this process. So as Einstein put it, make everything as simple as possible, but not simpler. And you need to express the evolution of these features and components, and maybe also their interaction together into mathematical equations. Then you need to put these equations into a computer through computer code and make the computer calculate a solution for these equations. Finally, if the assumptions you made in your model are correct, you would be able to predict the outcome for any given experiment. So, following this procedure, we studied first of all the permeability of the clots and the evolution of the flow during the lysis. So, experiments were conducted at the University Libre de Bruxelles, where fibrin clots were placed in tubes with physiological fibrin concentration, and a solution containing TPA was placed above these clots, and the solution was able to seep through the clot due to gravity. And at regular time intervals, the length of the clot was measured. So here on the right you can see a simulation of the model that reproduces this experiment, and with this model we were able to reproduce similar results as in the experiments, such as the fact that we have a clear separation between the interface between the clot and the solution, and thus that the position of this interface accelerated constantly in time. Also, we had a similar behavior of the flow restoration, meaning that we had a very slow increase in flow during most of the lysis and a very sudden increase towards the end of the lysis. So this is for the permeability and lysis conditions in homogeneous fibrin clots.
To further study how blood clot composition affects the effect of TPA on thrombolysis, we have been collecting ischemic stroke clots from the patients in the lab, and we have been investigating them under a microscope by histological techniques to learn about the composition, to learn about the internal architecture of these blood clots. And one of the main results or lessons from these studies was that these blood clots can be quite heterogeneous. Some clots are very platelet rich, whereas other clots are very red blood cell rich. And most of them have a mixed composition containing both of these components. And so the goal is in the lab to study these different types of clots on thrombolysis and see how TPA can lyse these blood clots. And we can do that on the patient clots themselves or use in vitro made clots in the lab. And when we make these clots in the lab, we can make these platelet-rich clots or make these red blood cell-rich clots. And the goal is to also model these clots via numerical simulations to mimic the experiments. And this shows how the modeling can mimic the experiments. On the left-hand side, you can see a graph showing the biological experiments where we can lyse these red blood cell-rich clots with TPA easily, and where the platelet-rich clots are more difficult to lyse using TPA, shown in blue. On the right-hand side, you can see the model itself, showing a blood clot containing blue parts, which is the platelet-rich parts, and red parts, which is the red blood cell-rich parts. And as you can see, the model nicely also shows that the platelet-rich areas are more difficult to lyse, and the red blood cell-rich areas are more easy to lyse, rendering lysis curves that are quite good, mimicking the real experimental data. So in conclusion, in this work package, I think it was very rewarding to combine the biological experiments with the modeling to solve another piece of the puzzle, to build in silico stroke trials.